The overall goal of this procedure is to examine the effects of small molecule inhibitors on fetal organ development using an ex vivo droplet culture technique. This is accomplished by first opening the peritoneum of a pregnant mouse and removing the embryo-containing uterus. Next, the embryos are removed from the uterus and are freed from the yolk and amniotic sacs. Then, the gonad mesonephros complex is dissected and isolated from the embryo. Finally, the gonad mesonephros complex is cultured in a droplet with or without a small molecule inhibitor. Ultimately, immunofluorescence microscopy is used to show changes in organ architecture using cell-type-specific antibodies.
So the advantage of our whole organ ex vivo upright droplet culture compared to, say, cell culture lines is that it has the advantage of having three-dimensional structure, giving rise to cellular interactions due to locality, as well as extracellular matrix signaling that may be important for organ formation and patterning. Furthermore, our upright droplet technique allows for less reagents to be utilized, therefore reducing cost and potential toxicity due to side effects. This is an advantage over other whole organ techniques, and it still allows for the diffusion of pharmacological agents to the target organ.
Although this technique can provide insight into the vascular development of fetal testes, it can also be applied to other fetal organs, like the lung, and we can also use it to investigate diverse biological processes or other cell signaling pathways during fetal development.